Well, thanks very much. I really appreciate the opportunity to give you just a progress report on this project that you all out there have funded. And so I want to especially thank right off the bat the very helpful families who've made this project possible. So, and I also want to start, sorry to switch the order of the slides on you a little bit here, but I want to point out that this whole project is really the brainchild of Sonia Valab and Eric Minical, together with their interactions with the ASO experts at Ionis Pharmaceuticals, including Holly Kordasiewicz, if that's, well, anyway, I'm not sure how to pronounce that, but Holly. So, and I want to say that most of the work that's been done so far has come from these people here. They've generated some really attractive looking ASOs that they hopefully soon will be shipping to Greg here in my lab for the experiments on prion-infected animals. So let me just give you a little outline, and Eric and Sonia prepared these slides, so I thank most of them anyway, and Holly some of the others, so I want to thank them for that. But the outline of the talk basically will mention what is a prion, and we've heard a lot about that already. And we've heard also a lot about targeting the healthy form of PRP as a potential therapeutic strategy, why to use this ASO or antisense technology, our progress so far, and our plans for the near future, hopefully. So you've seen a lot about this already today, so I won't belabor it, but in these diseases, the normal form of PRP is converted to an abnormal form that's usually clusters, highly organized clusters of PRP-SC. And as we've heard as well, one of the main strategies one might take in sort of preventing the disease process or affecting the disease process is to turn down the levels of the normal protein so that it can't be converted to the abnormal form. And one of the ways one of the types of evidence that this could be an attractive strategy is shown in this slide, where it's plotted, where we've plotted the time from infection to the time of first symptoms or terminal disease here as a function of how much PRPC, the normal form of PRP, is expressed in a transgenic mouse, and with 1 being the normal level of PRPC. And what you can see immediately is that mice that produce more than the normal amount of PRPC get sick and die sooner. And those that express less stay healthy longer. And we also know that mice that produce no PRPC never get prion disease. They're completely resistant to it. So this is one of the types of evidence that suggests that it might be worthwhile therapeutically to attempt to control the levels of PRPC in a person as well, if there were technology available to do it. And so we've heard already a bit about this question. It's a very important one. Is it safe to reduce PRPC levels? Well, there's some lines of evidence, at least, that it's, and we heard about this as well a little bit, that it may not be too damaging, but we don't have the final answer. But one thing we know is that mice engineered to produce no PRPC have only minor health issues that were hard to pick up at first. Mice engineered to produce half the normal amount of PRPC are indistinguishable from normal mice, at least in the types of tests that are done in the laboratory to assess such things. Not all possible things have been tested, of course. And then Eric Minical here has shown that humans with only 1 functional copy of the prion protein gene, that is, instead of 2, so they're presumably making half the amount of PRPC, the normal form, are healthy as far as we can tell. So our therapeutic hypothesis is that if we can make people produce less PRPC, they will stay healthy longer. And the question is, how might you do that? Well, one strategy that can be taken and has been taken already to some extent is to use this antisense technology. And so I'm sure you're aware that that proteins, including PRPC and PRPSC, have genes that encode them, and those genes are first transcribed to make RNA molecules, and then those RNA molecules are then decoded to make the protein amino acid sequence. And so this antisense technology attacks these RNAs, basically, and which, of course, will result in less production of a given protein, and they can do it very specifically. So how does this work? Well, the antisense molecules are oligonucleotides. They contain 20 nucleotides that have a specific sequence that binds to the RNA sequence that's meant to encode specific proteins, a very specific interaction. And when that interaction occurs, it encourages a particular digestive enzyme to come and destroy this RNA molecule, RNase H. And so it reduces the total amount of messenger RNA for that particular protein and reduces the amount of production of that protein, usually. And so different ASOs, or antisense oligonucleotides, have been developed against many different RNAs for many different encoding many different proteins and in being involved in many different diseases. So there's a bit of precedence here. And in fact, this technology is already in clinical trials for various neurological diseases. For instance, it's known that ASOs for brain disorders can be dosed into the intrathecal space, where the cerebral spinal fluid is at the base of the base of the spine, and you don't need to do it very often at all, maybe once every 4 months or so. And the patient's experience is a bit like undergoing a lumbar puncture to get or a spinal tap. And Phase 1 trials, that is initial drug safety trials for this spinal muscular atrophy, has found that this intrathecal delivery of ASOs can be safe, and they found preliminary evidence of efficacy, at least against this disease. And there's also and has progressed to Phase 3 trials, which are basically efficacy trials in large numbers of patients, say 1,000 patients or so. And there's also a Phase 1 trial for a Huntington's disease approach that was launched last fall, and the aim is to reduce the Huntington RNA levels. Huntington's the protein that gets misfolded in Huntington's disease, and with similar goals to ours for treating a prion disease. So, this antisense approach seems to meet all, or at least many, of the requirements to be a sound therapeutic strategy. It's known already to be basically, or appears to be, safe for chronic use. There's already one drug against a different RNA for a different protein that is already been approved and marketed for high cholesterol. And preliminary data from ASO trials for this spinal muscular atrophy indicate ASOs are well tolerated in the human brain. So that doesn't seem to be a problem at this point. And also, very importantly, it gets into the brain after this intrathecal delivery and can be stable for months after you inoculate them into the base of the spine, directly into the cerebral spinal fluid, which flows up and down the spinal column and around the brain and delivers things through the central nervous system. And so there seems to be good indication of good widespread brain distribution after this kind of single inoculation every few months or so. And the other thing that's attractive here is that it's likely that studies you perform in mice will likely be transferable to humans, at least in some fundamental senses, in that by targeting the RNA, the ASOs slow the disease process upstream of the generation, the immediate generation of PRPSC. As we've heard from in a couple of the talks at least before, that it may be desirable to affect PRPC levels, because then you don't have to worry about the strain-dependent effects that might be there for PRPSC and its interactions with drugs. And so we can avoid that, And by the same token, the strategy should be effective in multiple species and against all subtypes of prion protein that involve PRPSC generation, because all of them involve conversion of PRPC to PRPSC. So there has been previous work on ASOs for prion disease. In addition to targeting RNA, some ASOs we have shown in studies a long time ago that these ASO-like molecules, these small oligonucleotides that are metabolically stable, also have a profound ability to directly affect the conversion of PRPC to PRPSC without it being mediated by this antisense mechanism even. So there's two mechanisms by which these, fundamentally different mechanisms by which these ASO molecules might work. An ASO against PRP RNA was already found by this Prusner group to be at least moderately effective at delaying disease in mice. And now there have been some significant improvements in the ASO chemistry and sequence screening that have been performed by this Ionis pharma that seems to be leading to more effective, longer lasting, and safer ASO molecules that we now hope to test. And so the goals of our preclinical study were to develop new ASOs against the mouse prion protein RNA and to dose this directly into the brain with an intraventricular injection rather than the base of the spine. We want to test for the potency, that is, can these ASOs reduce the amount of PRPC in the mouse brain, the efficacy, how much can these extend the lives and the health of prion-infected mice, are they safe? And we also want to assess biomarkers that tell us what sort of progress we might be making, either in terms of reducing PRP-C levels or preventing PRPSC formation as predictors of efficacy. And the ultimate goal, of course, is to establish a proof of principle so that we can develop an ASO that against the human prion protein RNA and get it into clinical trials. So far, and again, practically all of this work with the ASOs, or all of it really so far, has been done at Ionis Pharmaceuticals. And they've screened 450 at least new ASO molecules in cell cultures, pulled out 26 of them as being most potent, then synthesized them on larger scales and so that they can be used in vivo testing. 11 of the most potent of these anti-PRP ASOs were selected for high-dose and long-term tolerability screenings. And 2 of the best of those have been used that have the longest duration and are best tolerated have been chosen for additional testing. And this plot here just shows in cell culture results at least these 2 final candidates, ASO1 and ASO2, if added to the culture of the medium of cultured cells, can really effectively reduce the PRP, the normal PRP messenger RNA levels by a substantial amount, 2 or 3-fold at least, both of these. And in an in vivo study, that is in animals and in mice, they've shown that a single bolus injection of these ASOs into the mouse, if you look 2 weeks later for effects on the normal PRP levels in the brain, both of them were quite effective at reducing from the normal level here down to a half or even less than half of the normal levels of PRP messenger RNA in the cerebral cortex of the brain or the spinal cord. So they've gone on to do another test in these animals. Again, no prions involved here yet, but with a considerably larger bolus injection, and then following these animals for 8 weeks post-treatment, again showing major decrease in the levels of the PRPC messenger RNA levels in both the brain and the spinal cord, without any signs of microglial activation, which be a sign of inflammation and some damage. There's no sign of that. in these animals, and there's no sign of any body weight change, which would be another broad indicator of problems due to these inoculations of these ASOs. So now at this point in time, I've already described that they've done a bit of work on the very important work, encouraging work on the potency of these ASOs, both in cells and in mice, and they're still involved in some retesting of these effects in animals before they send them to us for more safety and efficacy trials in our mice at Rocky Mountain Labs and in prion-inoculated mice at Rocky Mountain Labs. So that's going to go on per our grant proposal. Pretty soon, we hope. It seems like in the next week we should get started on that. We have already gotten an animal protocol approved through our Animal Use and Care Committee to do this work, and so we're primed and ready to go. And we also are going to look at this issue of biomarkers to determine whether these ASOs are causing reductions of PRP-C, the normal PRP levels in the brain, and we're looking to quantitate the levels of prion seeding activity, an indication of the presence of PRP-SC in animals that are either infected and either treated or not treated with the ASO, and then to establish our ability to measure PRP levels in the human cerebral spinal fluid as a potential biomarker and an indicator once we get to the point of hopefully doing this in humans, the efficacy, to be able to determine the efficacy of the ASOs that we might try there. So that's pretty much where we stand on this project. Debbie gave me couple more minutes to mention another project that we're pretty excited about on a separate issue, but I hope you'll find it interesting, and that is our discovery, along with this company, Briotech in Seattle, of a very mild but quite effective new anti-prion disinfectant called hypochlorous acid. And this just shows a picture of Dan Terry and Jeff Williams of the company standing in front of an aerosolizer here, being misted, showered with this hypochlorous acid solution that we now find is very effective in knocking out prions. So it's pretty innocuous stuff. And this is a collaboration between a large group of people in my lab, Briotech, and some people at the University of Washington as well. And just very briefly, I'll remind you that prions are really hard to kill. And so the main recommended treatments that are out there for decontaminating medical devices or tissues or whatever might be of concern are rather harsh, including very strong lye or sodium hydroxide solutions with very high pHs, very strong household bleach, also very basic, very high pH. And these usually together with, if possible, prolonged steamed autoclaving, which of course isn't possible for a lot of medical instrumentation and a lot of things. So there's long been a need for less toxic and caustic disinfectants for prions that might be applied to skin or any other parts of the body, to contaminated surfaces in hospitals and clinics and farms and meat processing plants, labs, you name it, and to decontaminate a variety of sometimes delicate medical instruments that are needed. And so why this hypochlorous acid, or this HOCl? Why were we interested in this? Well, it was originally identified as a broad spectrum intracellular germicide that's released by some of our immune cells, such as neutrophils, within the body to kill a wide variety of pathogens, bacteria, viruses, protozoans, fungi. And so this is well known to be quite effective and quite broad in its activity. And it's also, but it's related chemically to household bleach, but it's only a little bit acidic, about the acidity, say, of apple cider vinegar or something like that, rather than very strong, high pH, basic, caustic nature of household bleach. So it's chemically similar, but a lot less caustic. And its antimicrobial efficacy at least as far as I'm told, is about 100 times more effective than bleach. But unlike bleach, it's not cytotoxic. Even cells in the body are producing it, so I mean maybe it has to be contained and controlled well, but and it is environmentally friendly, breaking down basically into salt water. And it can be applied directly to skin and other surfaces, as I mentioned before. There are already some commercial preparations that are used in hospitals as disinfectants with about 50 to 100 ppm active chlorine, and the active chlorine content seems to be an important measure of the efficacy of these things. The Briotech proprietary production process produces typically materials about 400 ppm, so a bit stronger than this, and it seems to be more stable than the other preparations as well. So we call this Brio HOCl. And so, as I mentioned, just as proof of the innocuousness of this, it shows this stuff being aerosolized into an entire room and people able to stand there without even respirators while they're working with this stuff. And here's the president, Dan Terry, who figured out how to make this stuff, soaking his feet in buckets of this. So it's pretty innocuous stuff. Just as a last slide, I want to quickly summarize, though, our results in very cursory fashion here. And what we're finding in terms of prions is that this stuff can eliminate all detectable prion seeding activity as we measure by this amplification, very sensitive RT quick assay that I've described in previous conferences here, in infected brain tissue. And this applies to prions such as human CJD, variant CJD, classical BSE from cattle, sheep scrapie, and hamster scrapie. Like I say, it eliminates all that we can detect, which means, depending on which one we're talking about here, reductions of between 1,000 and 100,000. And this activity is comparable to the recommended bleach or NaOH treatment. And it also eliminated all detectable scrapie infectivity as measured by inoculation into mice. So it's not just this amplification assay, and indicated a reduction of at least 1 million-fold. And finally, just to look at what the situation might be for surgical instruments, which are usually often made of stainless steel, we coated stainless steel wires with scrapie infectivity and then treated them with HOCl and saw that it eliminated all detectable prion infectivity there. So, in addition, we've also heard already earlier at this meeting that there are all these other protein misfolding diseases that are resulting from amyloid seeding and propagation and so forth. So we're curious whether this is something specific for real TSE prions, or would it be applicable to these other types of self-propagating pathological protein aggregates. And we see strong reduction of the seeding activity of both tau, which is involved in Alzheimer's, CTE, and a number of other tauopathies, as well as alpha-synuclein activity. So we think that HOCl is an effective anti-prion reagent that is less caustic, less toxic, and more broadly applicable than other established anti-prion treatments. And we'd be really interested to hear from you out there what the applications might be, how you would need, how you need or want to use this sort of material in the various circumstances that you encounter. So I've mentioned the collaborators already, and I want to thank the families who have funded the ASO project. It's been extraordinarily helpful. So appreciate your attention, and I don't know if we're taking questions or leaving them until later. No, leave them until later. All right.